Okay, in today's lecture, what we're going to do is to begin to look more formally at the concept of information in living systems, which, as you know, is the organizing theme for the first third of this course. Now, in the second lecture, you'll remember, we discussed how one of the key problems to be solved concerning the origin of life was how to account for the way that a new trait that might arise is going to be preserved and, more importantly, transmitted to subsequent generations. To do this, we said, we need some sort of blueprint, a blueprint that somehow contains information about how to make other molecules. And we know that in modern cells, this blueprint is the molecule DNA. So DNA then represents molecular information, and we know that we'll eventually need to understand how information can be encoded in and extracted from this molecule. But first, before we look at the blueprint and how it works, what we have to do is look more closely at what that blueprint describes how to build. This means that we have to begin to understand something about the structure and function of proteins, which is the topic of today's lecture. So let's begin talking about proteins by looking at what they do. In Greek, the word proteos means first place, and this is a fitting origin for the name protein. It would be a little bit of an exaggeration to say that proteins do absolutely everything in cells. There are some things that proteins don't do, but it wouldn't be much of an exaggeration, and it's no exaggeration at all to say that proteins play at least some role in almost everything that cells and organisms do. Let me give you some examples of the kinds of things that these molecules called proteins do. They form structural elements, acting as supports to strengthen the outer wall of cells, or actually acting as little girders on the inside of cells, giving them their shape. They form what's called the cytoskeleton. That just simply means the cell's skeleton. As structural elements, proteins also help to hold together other large molecules that aren't as sturdy as they are. An example of this that's very important is DNA. If you look at DNA in a cell, it's actually surrounded by a whole series of proteins which are serving as a scaffolding to hold that DNA together. In fact, if you look at a chromosome, as we'll mention later, it's actually made of much more protein than it is of DNA, even though DNA is what's interesting about the chromosome. Some proteins actually perform mechanical work by acting as little motors. They cause cells to change shape by pulling on little elements inside. This is how muscles work, for example. Or they actually move materials around inside cells, acting as motors running little conveyor belts. Proteins actually act as the transport units themselves, serving as the vehicles that carry other materials around in cells or even around the body. An example of this is the protein hemoglobin, which is a protein responsible for transporting oxygen molecules in blood. The oxygen must bind to the hemoglobin, and that's the way that it gets carried around. Proteins also act as signal receivers. They act as receptors, taking in molecular signals, detecting signals within the cell or from other cells. And in fact, they often act as the signals themselves. Proteins act to regulate cell function. They actually act as little molecular switches, turning on and off the processes that a cell runs. Perhaps most importantly, proteins act as catalysts. We introduced this term before. That's just something that makes a chemical reaction run. We give proteins that act as catalysts a special name. We call them enzymes. And enzymes are especially important because they're what make the chemistry of life, what we call biochemistry, work at the temperatures and chemical concentrations found in living cells. So in a very real sense, proteins are the molecular workhorses of cells. The answer to almost any question about how something works in a living system invariably will involve some sort of protein at one level or another. This point, though, raises an interesting question. How can proteins do so many different things? Well, the answer is simply that there are many different kinds of proteins, each with its own unique function. For example, in a bacterial cell like E. coli, there might be 10,000 different proteins. In one of our own cells, there might be 50,000 different proteins, each one of these proteins specialized to do a different job in the cell. So, proteins are able to perform many different functions simply because there are many different kinds of proteins. But here's the next question. What makes a protein particularly good at doing whatever it does? How do these proteins become specialized for these very, very different functions? The answer to this question, and an important thing to keep in mind throughout this lecture and throughout the course, is very simple, really. The function of a protein largely is determined by the shape of that protein. Proteins are generally large molecules. Remember from earlier, they're polymers made up of a relatively limited number of amino acid building blocks strung together. But they're generally found in a very complex three-dimensional shape. The relationship between the shape and function of proteins is kind of intuitive for many of the things that proteins do. For example, let's say you're going to build a support structure, a molecular girder. You can imagine that the molecule that does this has to be elongated and has to be stiff. That's pretty straightforward. Similarly, let's take a more complex case, proteins serving as enzymes that catalyze reactions. Here as well, shape is really critically important because these enzymes will have a structure that includes special features, literally nooks and crannies on their surface, that will fit the other molecules with which that protein has to interact. And it's that fit that largely determines the enzyme's ability to catalyze a particular reaction. Now, it's actually a little bit more complicated than that because the chemical properties of the different part of this protein also matter in something like that. But the distribution of those chemical properties is largely a matter of the protein's shape. Well, so shape is what determines function, but this leads us to another question, and this question is getting us closer to this issue of information. What we know already is that proteins are polymers, long strings, of a relatively limited number of kinds of monomers, which are amino acids. So if a protein is just a string of amino acids, how do we get complex shapes to arise from that string? Now, to understand this, we have to delve into just a little bit of organic chemistry. Let's look at the building blocks, the amino acids. At the center of an amino acid is a carbon atom. This carbon atom is surrounded on three sides for all amino acids by the same three things. And then on the fourth side, it has something different attached depending on which amino acid it is. The three parts of an amino acid that are always the same are first a so-called carboxyl group. Now, a carboxyl group is just a chemical group that has its own carbon atom attached to one oxygen. It actually has a double attachment, a double bond to this oxygen, and then to another group called a hydroxyl group, which is an oxygen bonded to a hydrogen atom. So that's what a carboxyl group is, and that's one of the things that is attached to the central carbon of an amino acid. The second thing that all amino acids have in common is what's called an amino group. Now, an amino group is simply a nitrogen atom that's connected to two hydrogen atoms. The third thing that all amino acids have in common, one of the third bond that central carbon forms, is simply a hydrogen standing alone. Now, carbon builds four bonds. We mentioned that in the very first lecture when I introduced organic chemistry. This leaves one bond open. And it's the fourth bond that can be attached to any number of different chemical structures. This variable part of the amino acid is known as the side chain. It's also sometimes called the R group because when you're drawing a generic amino acid, you often would just put an R there instead of whatever particular chemical structure might actually be found there. There are 20 different amino acids that are found in living things, each characterized by a different side chain. Now, the side chain of an amino acid may be as simple as just another hydrogen standing alone. That actually is the simplest amino acid called glycine. or it may be its own more complex set of carbons, a carbon skeleton with hydrogens attached, or in fact, in more complex amino acids, it might have any number, well, not any number, but a number of other chemical elements attached, oxygens, nitrogens, and so forth. Now, for our purposes, remembering the details of amino acid chemistry isn't really important, and it's especially not really important to remember the differences between specific side chains for different amino acids. The important point is this, that the physical and chemical characteristics of the side chain of a particular amino acid determine that amino acid's particular characteristics, particular chemical characteristics. And this fact is going to become important later on because those particular properties, chemical properties of an amino acid side chain, contribute to the way that that amino acid in turn helps determine the protein's structure. Okay, now let's take another step. Remember, proteins are polypeptide chains. That means they're a polymerized series of amino acids, as we've discussed. I want to just look at how we get a polymer built of two amino acids. What happens is that the carboxyl group of one amino acid bonds to the amino group of another amino acid. Now, the details of this, again, don't matter. Really what happens is that a water molecule is formed when the hydroxyl of the carboxyl group joins with one of the hydrogens of the amino group. A water is lost, and then the nitrogen of the amino and the carbon of the carboxyl group are bonded together. There's just a couple of points to make about this that are important. The first thing I want to point out is to give a name to this bond, because I might use it later on. It's a kind of very strong chemical bond that's technically called a covalent bond. A covalent bond, once built, is very hard to break. And more particularly, for a chain of amino acids, that kind of bond is called a peptide bond. And hence we call a chain of amino acids a polypeptide. Now, the other important point to understand from this is that the resulting pattern in a chain of amino acids has what I'll call a backbone of repeated nitrogens and carbons. In other words, if you read along a chain of amino acids, element to element, molecule to molecule, you would see a nitrogen, a carbon, a nitrogen, a carbon, a nitrogen, a carbon. So you'd have nitrogen, carbon, carbon repeating. And that nitrogen, carbon, carbon repeating backbone is essentially the string that has been put together as we've polymerized a series of amino acids. Now, the reason this matters is that there is an inherent polarity to this string. There's going to be an open nitrogen on one end. All right, this is the so-called N-terminus of an amino acid chain, and there's going to be an open carboxylic acid on the other end. This is the C-terminus. I should have said that there's going to be an open amino group on one end and an open carboxylic acid on one end, on the other end. So an N on one end, a C on the other. So there's an inherent polarity to the string of amino acids. This polarity will become important later on when we learn how DNA can actually form a code for this string of amino acids, because it determines which direction the code has to run in. Okay, now proteins can be variable lengths. The length of a polypeptide in a protein can be quite short in some cases, maybe only 50 amino acids long, or it can actually be quite large. Some large proteins may be 5,000 amino acids all strung together. An average protein may be, say, several hundred amino acids long. That would be typical. Okay. Now we come to the next important question. How does the unique sequence of amino acids, which we now know will have some unique pattern of side chains running along that sequence, how does that determine the function or the structure of a protein? And the answer to this is the fact that these amino acid chains, once they're formed, once they're polymerized, don't remain stretched out. Instead, what happens is they inherently begin to fold up, to twist back on each other, and to get all bunched up in more of a globular form. And this is the important thing. The way they fold together is not random. but depends on the sequence of amino acids. So the remarkable fact about proteins is that the unique three-dimensional structure of any protein is specified by the linear sequence of its amino acids. So how can that be? How does that work? Well, let me explain that. You see, when the polypeptide is formed and the chains fold spontaneously, what makes it fold in a particular way has to do with the way that the side chains of the particular amino acids interact with each other physically and chemically, and also the way those side chains interact with the environment that the protein finds itself, which in living systems is usually some sort of watery environment. So it's these interactions between the side chains of the amino acids along the sequence with each other and those side chains with the external environment, if you will, that are what determine a specific characteristic three-dimensional shape for that protein. Let me clarify this now by looking in a little more detail at protein structure. Okay, and the point of this is to understand how sequence of amino acids leads to a particular structure. But I'll do this by laying out what are the four levels that we can describe for a protein structure. These four levels, by the way, when we're talking about protein structure, themselves form a hierarchy. As we've seen, a hierarchy as a hierarchical organization is very important in living systems. And here, even on the level of molecules, there's a hierarchy of structure within a single molecule. These levels are named somewhat simply the primary level of structure, the secondary level of structure, tertiary level of structure, and the fourth level, which we'll get to at the end, is the quaternary level of structure. So let's look at each of these in turn. So a protein's primary structure refers to what we've already discussed, which is simply the sequence of amino acids that make up that protein. So for example, we would be able to describe the primary structure of a polypeptide completely by just giving an ordered list of the amino acids that comprise it. So we might give a list that would be, for example, valine, glycine, serine, leucine, proline, glycine, glycine, valine, and so forth. Early work on protein biochemistry really focused heavily on determining what the primary structure of proteins were before people were able to start looking in more detail at higher levels of structure. For example, the very first protein that had its amino acid sequence completely sequenced was done in the 50s. It was the protein insulin, which is a hormone, a kind of molecular messenger that in this case is involved in the regulation of blood sugar in vertebrates. This was sequenced by Frederick Sanger at Cambridge University, and it was Sanger's work with insulin which first demonstrated something that biologists take for granted today, but which wasn't known then, and that is that each protein has its own unique sequence. Every different protein you look at will have a different sequence of amino acids, and this sequence of amino acids is its primary structure. The secondary structure of a protein refers to the first level of bending and folding that occurs, the first level of three-dimensional shape that emerges. And it actually emerges as an emergent property of this sequence of amino acids. Now, the secondary structure we're not going to dwell on very much right now. It's actually very important to protein biochemists and was early on when they were trying to figure out how proteins were built. And the reason is because these are kinds of structures that are very common to parts of different kinds of proteins. These two levels of structure or kinds of organization are called respectively an alpha helix, which was described by Linus Pauling, and the beta pleated sheet. Now, these particular structures come about from weak chemical interactions that are occurring along the backbone of the amino acid sequence. Remember, the carbons and the nitrogens that are the backbone form a very regularly repeating pattern of nitrogen, carbon, carbon, nitrogen, carbon, carbon. And those nitrogens and carbons and the regular unvariable part of the amino acid can form weak molecular interactions with each other that we call hydrogen bonds. Now, I've already defined for you a covalent bond, which is a very, very strong chemical bond that once built is hard to break. Hydrogen bonds, by contrast, are very weak chemical bonds. They actually can be built and dissolve very quickly. In fact, it takes a lot of hydrogen bonds, or at least several hydrogen bonds, to hold different parts of a molecule together, because one hydrogen bond alone is pretty weak. Now, hydrogen bonding along long stretches of an amino acid sequence, all taken together, can hold that sequence in particular patterns like an alpha helix or a beta pleated sheet, simply because the regular pattern of the amino acid backbone provides enough common opportunities for hydrogen bonding to occur that leads to these higher level three-dimensional structures. Okay, now I don't want to dwell on secondary structure for its own sake, because what I really want to get to is what's called the tertiary structure of a protein. And this is the level of structure that we're most interested in, because it's at this level of structure where we begin to see how structure determines the function of a particular protein. The tertiary structure of a protein is also the result of chemical interactions that occur among the different amino acids in the string. But in this case, these are interactions between the side chains that are variable, as opposed to the carbon-nitrogen backbone. Now, the fact that the side chains are occurring in an irregular sequence, or can occur in an irregular sequence. In other words, every different amino acid along the sequence may have a different side chain, means that the pattern of interaction among those side chains, the pattern of chemical interaction, can be quite variable, much less regular, and much less predictable. What that means is that the pattern of twisting and folding that results from these interactions that cause tertiary structure become much more complex, not regular like the alpha helix or the beta pleated sheet, but actually in some ways quite unpredictable. These interactions involve three basic kinds of ways that these side chains will interact with each other and with the environment around them. The first kind of interaction stems from the fact that different side chains of different amino acids are more or less willing to be near water molecules. Let me give you some terms that we can use here. Some amino acids are what we would call hydrophobic. Literally, that means water-hating. That means that the side chains of these amino acids don't play well with water. They're actually repelled by water. Now, other amino acid side chains are what we would call hydrophilic, literally water-loving. And these, therefore, will be attracted by water. Now, remember, proteins almost invariably find themselves in a watery environment. So what happens is that those amino acids that are hydrophobic will tend to move and twist and turn to get away from water. And what that means is that those amino acids will find themselves moving in towards the center of what is going to emerge as a globular protein, because that's where they're the farthest away from the water that surrounds the protein. On the other hand, the hydrophilic amino acids do just the opposite. They will tend to move and twist and turn so that they find themselves on the outside of the protein. So it's the watery environment that proteins find themselves in living systems that causes these amino acids that vary in how hydrophobic or hydrophilic they are to twist and turn. Now let's step back and remember that a protein is going to be a variable sequence of different amino acids, some more hydrophilic, some more hydrophobic. Depending on that sequence, depending on the particular arrangement of amino acids, you're going to have different combinations of forces on that string to bend and twist in different ways to get some amino acids on the inside, near the inside, some amino acids more towards the outside. So this is a major determinant of what causes a protein linear structure to bend and fold, not just bending and folding, but bending and folding in a particular pattern to maximize contact or minimize contact with water on the outside. Now, there's a second kind of interaction among the side chains of different amino acids which also contributes to this bending and folding. In this case, it's also it's a set of weak chemical interactions, including hydrogen bonds, that are formed between the chemical structures of different amino acid side chains. We've already seen hydrogen bonds important in the secondary structure of proteins, and in this case we also can find, in some cases, hydrogen bonding pulling one amino acid a little bit this way towards another amino acid that's pulled this way so that they get a little bit closer together to form some kind of hydrogen bond or other weak chemical interaction. Now, this differs from the hydrogen bonding that we saw when we were talking about secondary structure because, again, the pattern of amino acids in the sequence is going to be variable, not predictable like the backbone of the amino acid. So we don't get regular patterns like alpha helices or beta-pleated sheets from this kind of weak chemical interaction, but instead this contributes, like the hydrophobic and hydrophilic nature of amino acids, to these different amino acids in a chain trying to get a little closer or a little farther apart from each other. So we have two sets of forces, trying to get a little closer or a little farther apart from the water that surrounds the protein, and then trying to get a little closer or a little farther apart to the other amino acids. Imagine a third grade class which has to go out on a field trip. Let's say, I don't know exactly where they would be going as a field trip, but let's say they're going to the beach. And some of the kids like to be near the water, and some of the kids don't like to be near the water. You might be able to predict then that when you see this class of third graders running around, that some are going to be more likely to be near the water, some farther from the water. And so what you see then is you can predict what the distribution, what the shape of that third grade class is going to be. In addition, some of the kids like each other more than others. So in addition to whether they're near the water or farther than the water, friends are going to be a little bit closer to each other, and people who aren't friends are going to be a little farther from each other. This is sort of a way to think about how the particular chemical properties of amino acids affect its shape. Now the difference is that you don't have a third grade class all tied together in a string. Well, I guess on some field trips you might, but ordinarily you wouldn't be doing that. Now there's a final set of factors that contribute to the three-dimensional shape of a protein, and this factor has to do with stronger chemical bonds that can be formed between particular kinds of amino acids. These are actually covalent bonds. Particular amino acids will covalently bond with each other. That again pulls certain amino acids closer to each other than others, and furthermore it greatly strengthens and stabilizes the three-dimensional structure of a protein. Okay, so for a single polypeptide, that is a single chain of amino acids, the three-dimensional structure is really the highest level of structure for a protein. Now, I just want to mention briefly that there are some proteins that actually have what we call a quaternary structure. And this simply refers to the fact that these proteins are made of not just one polypeptide chain, but several. Hemoglobin here is an example. This protein actually has 4 different polypeptides, 4 subcomponents that all bind together. And it's only when these different polypeptide subcomponents not only fold with themselves, but also fold into each other, that you have the full structure of the protein. And we call that quaternary structure. Now, the most important take-home message from this lecture on protein structure is simply this: that if you've got a string of amino acids of a particular sequence, this will lead to a particular three-dimensional structure. That means the shape, and therefore the function of a protein, all boils down to the order of amino acids in a chain. So if we're interested in how information can be encoded in a blueprint for building proteins, what it means is that all we simply need is to have information about the order of amino acids in the sequence of that protein. Because if we've got the sequence, the structure emerges on its own. Contrast this to a blueprint, say, for building a house. It would be like you'd be able to build a house if you were given a list of the parts that you needed for that house in the order that you'd happen to use them. You know, a 4-foot 2x4, okay, a nail, now another nail. I mean, this would be a really crummy blueprint for building a house. But it's exactly this that's the blueprint, or that can be the blueprint, for building an amino acid. And in fact, a polypeptide chain or a protein. And in fact, a protein is much, much more complicated, as you can imagine, than a typical house. Beginning in the next lecture, we'll ask exactly what this blueprint is in cells, what it's made of, and how it really works.